Okay, let's look at viruses. First of all, viruses are interesting. Um, if you sit there and classify living organisms, there is not a classification for viruses because they are not living. They are not found on the tree of life. They have genetic material, but they can only multiply that if they find a living host cell. So they're not a live organism. They are a parasite, what we call an obligate intracellular parasite. They have to be inside a cell in order to do anything. They need that cell for energy. They need it to get metabolites. They need it for protein synthesis. They do have their own nucleic acid genome. You have RNA viruses and DNA viruses, and we'll look at this. But these are not living organisms, but they sure as heck can cause disease. Okay. The extracellular form of the virus is called a viron, and it exists outside the host, and it helps transmission from one host cell to another. Reproduction, replication only occurs, as it says, upon infection and entry into a host cell. So in order for the virus to divide, it's got to get inside a cell. Now, these guys do cause many, many, many infections, humans, animals, plants, bacteria. They can even infect bacteriophage. They cannot carry out any metabolic pathway. They use their host. They can't grow, they can't respond, they can't reproduce. And if you sit there and look at what's on that slide, carry out metabolic pathways, grow, respond to the environment, reproduce independently. Those are things that determine for us as biologists, is this a living organism? And this is what makes our virus not classified as a living organism because it doesn't fit what we think of as a living organism. All right, so when we look at these viruses, they do cause most diseases that give us all kinds of problems in the industrialized world. A virus is a small piece of either DNA or RNA or pieces, okay? There is no membrane, there is no cytosol, there is no organelles, there is an exception, but they do have an extracellular state and an intracellular state. So they have where they are outside the cell and where they are inside the cell. So first of all, let's look at them outside the cell. They are again called a viron. They have a protein coat that surrounds their nucleic acid. And they have a capsid, and so that's called a nucleocapsid. Some of them are enveloped, and that envelope will be a phospholipid, and we'll talk about this. And we've talked, if you got an envelope, it makes it fairly easy to kill because, again, once you remove the envelope, the virus is dead, as we mentioned before. So it's the outermost layer that provides recognition sites that allow it to attach to the host cell. It also protects the virus. The intracellular state, the capsid is removed, the protein coat is removed, and the virus exists only as a nucleic acid. So if we looked at this envelope virus, you can see this is an RNA virus because that's the nucleic acid inside. And this is the capsid, all right? And this is the lipid envelope. This is an envelope virus. And you can see this hemagglutinin and neuraminidase. FYI, we'll talk about this, but have you ever heard about the H1N1 virus or H1, N1, H3? This is what it means. This is the hemagglutinin and the neuraminidase. And that's how we classify these viruses, by the types, these two types, okay? So how do we distinguish them? The type of genetic material they contain. Look at this. Are they a DNA virus? Are they an RNA virus? They may be a double-stranded, okay? The kinds of cells they attack. Are they animal viruses? Are they plant viruses? The size of the virus. How big is it? What does the capsid coat look like? The shape of the virus. I've got a picture coming right next. Is a helicovirus. Is it an envelope virus or a non-envelope virus? So when we look at viruses and start classifying, we want to look at all these things, okay? So here's a helical shape. Here's a polyhedral. This is a very complex shape. And this guy is a T4 phage. This is a guy that actually attacks bacteria. All right. So when we look at the size of viruses, they're very, very, very small. Okay, so here you have a red blood cell, and next to it is our lovely E. coli. It's tiny, 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 tiny little dots are the viruses. Okay, so these are very, very, very small. We cannot see them underneath a microscope, a light microscope. You need an electron microscope to look at these things, okay? We can certainly see the red blood cell. We can certainly see the bacteria, although they are small underneath the microscope. But when you start getting here, yeah, you need a light microscope, electron microscope, sorry. And if you look at this, some of these are very small. The polio virus, very tiny, versus smallpox virus, a lot bigger. So it all depends. Some of them are very tiny. This one is infecting bacteria, a little smaller. Then you got some bigger ones here. Okay. So as I mentioned, RNA and DNA, they do show a lot of variety in their genome. They can be DNA or RNA. They are never, ever both. They'll pick one. And we classify them, and this is the way we primarily classify them. They can be double-stranded DNA viruses, single-stranded DNA viruses, double-stranded RNA viruses, or single-stranded RNA viruses. They may be linear or composed of segments or circular. So it gets pretty interesting. The other way we classify them, as I mentioned, is the kind of host. Most of these viruses only infect a particular kind of host cell because they have to be able to bind to it. They have an affinity that's complementary to the proteins and the glycoproteins on the host cell. So if you're trying to get inside a human cell, you got to have surface proteins or glycoproteins that fit in your host cell so that you can attach. So some of these guys may only infect a particular kind of cell in the host. It may be that particular. Or you may have generalists that can infect many different kinds of cells in different hosts. So it all depends on a particular virus. This is what a virus can do. This is a plant virus, tobacco mosaic virus, specific to tobacco. And you can see the nice, we grow tobacco in North Carolina. So here's your lovely little tobacco leaf, and here it is infected with tobacco mosaic virus. Okay. This is an electron micrograph of a phage, bacteriophage, attacking a bacteria. All right. All right. Then we have the capsid. This is the protein coat that provides protection for that nucleic acid, whatever one it is. And it's how the virus can attach to the host cells. It is composed of protein subunits called capsomeres. And again, you may have a naked virus or an envelope virus. Okay? Naked viruses, they don't have that outside envelope. Most bacterial and plant viruses are naked virus. They have no other layer. It's just the nucleic acid and the capsid. Then you do have envelovirus, which many of the animal viruses are, and they do have an outer layer that has a phospholipid bilayer from the host cell. We'll talk about it. So when they get released from your cell, they are released with your own cell membrane, which makes it harder to find them, and viral proteins. And the nucleocapsid is that nucleic acid plus the protein in the enveloped virus. So if we look at this, you can see the naked virus. There's that capsid. There's this nucleic acid. Whereas the envelope virus has that lovely little envelope. And this is, again, for humans, a lot of that, this is your actual cell membrane. We'll get to that. All right, so as I said, the viral envelope is acquired from the host cell during replication or release. And the envelope is actually a piece of the cell membrane of the host. Okay, so you get a phospholipid bilayer and proteins, and some of these proteins are virally coded. They're spikes. The envelope's proteins and glycoproteins play a huge role in host recognition because it's very difficult for your immune system, just FYI, to recognize, is this a virus, envelope virus, or is this my cell? It makes it hard, okay? All right, when we replicate viruses, there are cycles, the lytic cycle and the lysogenic cycle. Sound familiar? So the core difference is the lytic cell obviously causes the cell to lyse, but it immediately, when it gets a hold of that cell, it destroys the host cell to spread copies of that virus. Okay, when it goes through that finish, it's going to hijack the cell, force it to produce more viruses, and then release them out. The lysogenic cycle integrates the viral DNA into the host genome to replicate passively without harming the cell. The viruses that can perform both of these cycles, which are called temperate phages, typically prefer the lysogenic cycle when the host cell is stable and resources are plentiful and everything is good and happy. It'd rather just sit there and reproduce. However, if there's some kind of environmental stress and, it's not going to look like it's going to survive, the virus can, quote unquote, wake up, excise its host genome, and go into the lytic cycle, which throws its genome out there, if you will. So, the virus is dependent on the host organelles and enzymes to produce new virons. Again, this cycle usually results in the death and the lysis of this host cell. So when we go into this lytic replication cycle, I'll show you in a second, there are several stages. You have to attach, you have to have the entry, then you get the synthesis of the nucleic acids and the proteins because it's redirecting the host cell to do this, and then it assembles it and packages these new viruses, and then we release them. Now, the major difference between prokaryotic and eukaryotic viruses is the nucleic acid entry between the two. All right, so let's kind of look at this. Here we go. So first of all, we have to attach. Coming in, we're attaching here, and this is a bacteriophage, so it's attaching to a bacterium. So it will attach. It has a sheath, and what happens? That's the attachment phase. There's that little circular bacterial chromosome that we talked about before, and it will penetrate through that cell wall. Okay. And once that happens, then you get the phage DNA begins to be replicated. the bacterial chromosome degrades, and the phage takes over and uses that mechanism to make new phages. So it makes all the little pieces of the phage, they get assembled. Once they've got enough made, the cell lyses. Okay? And you can see in this assembly, it's got to make all these different parts until you get everything that that viron needs to be released. Okay? Typically, in a lytic cycle for bacteria, it doesn't take too long. Okay? You can see early enzymes are made, the nucleic acid ready, the protein coat is made, and then the assembly and release, and boom, you're done. Animal viruses, a little different. But the major tenets, major manufacturing, the takeover of the host, the assembly and the release, and everything like that is universal. Eukaryotic viruses are classified by the genome. Again, is it DNA viruses, RNA virus? There are 3 differences. When that viron enters the cell, the entire viron enters the cell. Okay? Because we have a cell membrane, we've got to bring it into the cell. The eukaryotic cells contain a nucleus, which is the site of the replication for many of these animal viruses. And we basically get a membrane-bound viral factory, viral plasms, that form in some of the eukaryotic cells to increase the assembly rate and protect from the host defense. So, again, it's basic same replication pathway, but you have to realize the presence of the envelope around some of the viruses, the lack of the cell walls in the animal viruses, and basically that we're a eukaryotic cell, we have a lot more those little things like mitochondria and stuff like that. Okay, so it does make a difference. We do have organelles that the bacteria don't have. So first of all, we got to get attachment, and this is a chemical attachment. And again, animal viruses do not have tails or tail fibers or anything like that. They do have spikes or molecules that will help it attach, okay? And then what happens, so you've got a receptor on the cell, and it will touch it and match it, okay? And you can see that. And what happens is it cuts, this is the cut edge of the host cell. You can see it, this is cut through. So you see the genome is here. It's not like we saw with the bacteriophage. Let me go back here real quick, where you just got the tail fibers coming down. Okay, you can't do that with an animal cell. So what happens is that only the genome goes in. Okay, that's direct penetration. For most of these, what happens is that particularly in the virus, the enveloped viruses, it will touch your cell membrane. And just like, if you remember in biology, endocytosis, when you bring something into a cell, you bring it in by touching that cell membrane, and it wraps itself around it, and then it brings it in. So exact same thing happens because this is coated with our glycoproteins that are our glycoproteins, because that's how we're going to get made. So it touches that membrane. It allows for this opening up, and then the viral genome comes on in, and the whole virus, and it gets uncoated, which releases that genome. Okay? So each type of animal virus requires a different strategy depending upon its nucleic acid. So most of the DNA viruses assemble in and are released from the nucleus into the cytosol. Most RNA viruses are just replicated in the cytoplasm. There are a number of viruses produced and released. The number depends on the type of virus, the size of the virus, and the initial host of the host cell. If it's a good, healthy host cell, it can get going. If it's sick, it doesn't do as well. Envelope viruses can cause persistent infections, again, because it's wrapped in your own cell membrane, and it makes it much more difficult for our immune system to distinguish. Naked viruses released by exocytosis and may cause lysis and death of the host cell as it exits. Okay? So here's your little viral capsid that's been replicated now by your host cell. And as I said, it touches the cytoplasm, it wraps it in our cell membrane, and it buds off. And now we have an enveloped viron that's going to go off and do the same thing and replicate and replicate and replicate. Okay? So animal viruses can be dormant for years. They can remain dormant in the host cell. It may be prolonged for years with no viral activity or signs or symptoms. And some of these latent viruses don't become incorporated in the host genome. Some do. And we'll look at that. But think of something like some of these diseases that you may get where you get symptoms later. Okay. Chickenpox goes and harbors in the nerves. And years later down the road, you come down with what? Shingles. Okay, so this is an example. So let's look at these guys. This is just a representation of some of the human viruses, and you can see whether they're double-stranded or single-stranded, or a retrovirus. We'll talk about what that is, and how big these are. So the herpes virus, pretty big virus versus the HIV, not very big. Okay, so let's look at this. When you have an animal viron assembly and it's finished, you have 4 outcomes that can occur. One is you're going to have a virulent infection. This is the most common. You lyse the host cell, and that virus is released. You can have a latent infection where the viral DNA exists in the host genome, excuse me, as a provirus, okay, and virins are not produced. The host cell is unharmed until a virulent pathway is triggered. Okay, and then you can get persistent infections. Very slow release of the virons from the host cell by budding. Does not really, like we just saw, doesn't result in cell lysis. The infected cell remains alive and continues to produce virus. Okay, and then we can get transformation, which is a conversion over from a normal cell into a tumor cell, as in a cancer virus. So you can take a look at these pathways. I kind of described it for you. Okay, so here's your virus in your cell, and you can get tumor cell division and get transformation and form a cancer. You can cause the death of the cell and the release of the virus, lysis. Okay, you can get a persistent infection because you have a slow release of the virus causing cell death, or you can have a latent